नमस्कार कृषी मित्र आणि मैत्रिणींनो. आज आपण नॅशनल एलिजिबिलिटी टेस्ट नेट 2025 यामध्ये ARS, SMS अँड STO एक्झाम यासाठी लागणाऱ्या प्लांट पॅथॉलॉजी या विषयासाठी लॅबमध्ये जे इन्स्ट्रुमेंट्स यूज केले जातात त्याबद्दल माहिती घेणार आहोत. Acquaintance with laboratory equipments, instruments and tools. In that, first one is equipments. First equipment is spirit lamp. In that, first point is metallic glass lamp in which rectified spirit is used as a fuel. Second one, used for sterilization of metallic articles. Examples: inoculating needle, loop, forceps, scissor, spatula, mouth or test tubes, flask, glass slide, glass rod, etc. by direct flaming on flame temperature greater than 200 degrees Celsius. Second instrument is very important, that is hot air oven. In that, dry hot air at 160 degrees Celsius to 180 degrees Celsius minute is used for sterilization of glasswares for petri dish, flask, beakers, test tubes, pipettes. Oil, waxes, powders, rubber or plastic articles, soil compost, culture media, etc. are thermosensitive, so should not be sterilized in a hot air oven. See this picture. This is the hot air oven. In that we sterilize the glasswares. Autoclave. Third one, very important instrument which is used in pathology lab. In that, saturated steam under pressure is used for sterilization of the materials like culture media, solid or liquids, for killing, destroying cells, spores of the organisms. Autoclave means it is same like cooker, pressure cooker. Fourth one is laminar airflow cabinet, sterilization chamber also called. First, used to prevent contamination of pure cultures of organisms with unwanted microbes. Second, in this high frequency particulate air, filters are used to maintain microbes free air in working chamber, and it is sterilized with UV tubes. Third point is it provides aseptic conditions for isolation of microorganisms. Fifth, most important instrument is biological oxygen demand incubator. It is closed metallic chamber which is used to incubate culturing of microorganisms at controlled temperature and conditions favorable for the growth of and development. With fluorescent tubes, light required for sporulation and growth of microorganisms also provided. Sixth very important instrument is cubic colony counter. In that, first point is it is an electric apparatus used to count the number of colonies of bacteria or other organisms grown in culture plate. Seventh one is pH meter. Most important instrument is pH meter. This is small. It is used to determine pH acidity or alkalinity of solution and culture media, as well as setting of the pH of various media or solutions. Eighth one is centrifuge. It is an apparatus that rotates a high speed and separates substance particles, suspended matters, microorganisms on the basis of mass and density by means of centrifugal force. Various types of centrifuges commonly used are low speed clinical centrifuge, speed 500 rpm, a very high speed 1800 to 2000 rpm, and super ultra centrifuge, it is 2000 to 6000 rpm. Ninth instrument is spectrophotometer, that is called colorimeter. It is an instrument used for counting bacterial population based on the principle of turbidity. In that, the amount of light absorbed and scattered is proportional to the mass of cells in the light path. Turbidity of nutrient broth increases as the number of bacterial cell increases. 10th, refrigerator, that is fridge, very important in every lab. It is the basic equipment required in microbiology lab, used for storing, preserving stock, pure culture of microorganisms at 4 degrees Celsius. At this low temperature, microorganisms remain inactive and do not suffer from damage due to evaporation of the medium. It is also used for storing serum, antibiotics, and biochemical agents. Third one, second point is tools. In that, first inoculating needles, that is called wire loops. It is the most commonly used tool for aseptic transfer and handle of microorganisms. It consists of an insulated handle with screw device which holds a heat resistance nichrome or platinum wire approximately 3-inch long. Loop is used for transfer of liquid culture and needle for microorganisms culture growing on solid media. Inoculating needle loop is sterilized by heating on flame of spirit lamp Bunsen burner. Second tool is hand lens or magnifying lens. It is a very small single lens, simple microscopic used magnifying and observe plant diseases specimens. Its magnifying power is 10x. Third is washing bottle. Plastic bottle filled with water and used for washing gently the excess stain on glass slide during simple staining of bacteria. Third is glasswares. Many types of glasswares are used to sterilize in that the hot air oven. That is example is petri dish, conical flask, beakers, culture test tubes, glass slide, glass rods, funnels, graduated cylinders, graduated pipettes, reagents, glass cover slips, etc. are used to examine or preservation for the microorganisms. Last one is culture media. For growing microorganisms in lab, various types of culture media medium are required. In that, natural empirical, that is milk, urine, diluted blood, vegetable juice, infusion meat extracts are used. Second type is semi-synthetic. In that, potato dextrose agar, yeast extract, mannitol agar, blood agar, mac cornicus agar, sub or God agar, Richard's agar, glycerol yeast extract agar, Martin's rose Bengal agar, etc. are used. Last one is miscellaneous. It is required in microbiology lab, non-absorbent cotton, stains and staining reagents, disinfectants, distilled water, butter papers, blotting papers, filters, papers, rubbers, bands, muslin, cheese, cloths, gas stove, mixture, cum grinder, brush, glass marking pencil, sticker, scales, knife, razor blade, etc. I hope this video is useful for this NET exam 2025 for all agriculture students in plant pathology discipline. Thank you so much for watching this video.